DNA that exists in the nuclei of your cells is present in just one copy per cell, in the case of the X or Y chromosome in males, or 2 copies per cell in the case of the 2 X chromosomes in females, and chromosomes other than the sex chromosomes. In the laboratory, it is very difficult to manipulate or visualize a sample containing just 1 or 2 copies of a DNA molecule. Instead, researchers typically work with many identical copies of the DNA molecule that they are interested in. A common method for making copies of a piece of DNA is the polymerase chain reaction, or PCR, which allows a targeted region of a DNA molecule to be replicated or amplified into as many copies as desired. PCR is both selective and highly sensitive, so it is used to amplify and detect small quantities of nucleic acids, such as HIV in blood bank supplies, or to study DNA samples as minuscule as those left by a smoker's lips on a cigarette butt dropped at the scene of a crime. The starting sample can even be as small as a single molecule of DNA. In this video, the principles of PCR are illustrated. Because the PCR reaction is essentially a DNA synthesis reaction, it requires the same basic components used by the cell to replicate its own DNA. In this case, the procedure takes place in a small plastic tube containing a solution that includes 4 essential components. The first of these is template DNA. At least one molecule of double-stranded DNA containing the region to be amplified serves as the template for amplification. The second component is DNA polymerase. The enzyme DNA polymerase is used to replicate the DNA. The third component is all 4 deoxynucleoside triphosphates. Deoxynucleoside triphosphates with the bases A, T, G, or C are needed as building blocks for the synthesis of new DNA strands. The fourth component is 2 primers. 2 short sequences of single-stranded DNA are required for the DNA polymerase to start synthesis. Enough primer is added so that the number of primer DNA molecules is much greater than the number of template DNA molecules. The primer sequences are oligonucleotides produced by chemical synthesis and are typically 20 to 30 nucleotides long. Their base sequences are chosen to be complementary to the ends of the region of template DNA to be amplified. In other words, the primers flank the specific region of DNA to be amplified. The 3' end of each primer must be oriented toward the region to be amplified, so that when DNA polymerase extends the primer, it creates a new DNA strand complementary to the targeted region. Because the 3' ends of the primers both point toward the targeted region, one of the primers pairs with one of the template strands and the other pairs with the other template strand. Your task here is to choose primer sequences that are complementary to the ends of the target DNA sequence.